Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host.
Dr. Erlich, thank you so much for coming back on the show. We had such a great conversation a few episodes ago about your book, The Genetic Reconstruction of the Past. And for people that are seeing this on video, you can see that I really have read it because you can see how many notes I have. And that's just when I was physically reading. I went from this to my Kindle copy, depending. So we were able to talk about the first half of the book where you focused on how PCR has really changed criminal cases and that whole aspect. But then the second half of the book, we didn't get to talk about. So I was like, let's just have you back on because you more focus in the second part of the book about how PCR has enabled us to really look at evolution and ancestry of humans, but also even before that and different species. Tell me a little bit more about how in the last 30 years, we've learned so much, particularly in these last 3 decades, about evolution and how species are related. How exactly did PCR amplification help with this? Because that's really what enabled a lot more research and understanding to happen 30 years ago.
Well, the idea behind the book, The Genetic Reconstruction of the Past, was that these two apparently disparate fields, forensics and the criminal justice system and the study of evolution, really have a lot in common. The analysis uses the same kind of technology, PCR amplification and DNA sequencing, and the inferences that we make in both areas, whether the criminal evidence matches the suspect or how we can reconstruct human migrations and population movements, it's really dependent on exactly the same kind of data. So, for instance, if you have a genetic variant, you may want to know what the frequency in the population is. So, for instance, to interpret a match between the crime scene evidence and a suspect, that means the genetic profile is the same or indistinguishable. But to interpret that, you have to know the frequency of that profile in the population. So that's what's called population genetics databasing, the frequency of different genetic profiles in a given population. And if you want to reconstruct human migrations and look at human evolution, and let's say you have 3 different populations, A, B, and C, and you want to know how they're related, you look at the distribution of genetic variants in population A, B, and C, and if what's called the genetic distance, that's a metric that measures the differences between populations. If that's smaller between A and B and A are further from C, you can say that the A and B are closely related, but they're distant from C. And so you can reconstruct human migrations, that is, by looking at the genetic relationship of populations using the distribution of genetic variants in the population. And of course, that's what's also used to interpret a match. So that was the idea behind the book, that these apparently very different fields have this commonality. And the other commonality that I alluded to in my title is that you're reconstructing history. You're looking at the past. In part 1, where we look at the crime scene, you're trying to reconstruct the events that happened at the crime scene, so the very recent past. But in human evolutionary studies, you're trying to recreate events that happened in the ancient past. And I guess another commonality is when you're using PCR to amplify crime scene evidence, often the DNA is degraded, that is broken up into little bits. And so it's a technical challenge to amplify and analyze that, but PCR enables you to do that. And the same thing is true of many evolutionary studies. You're looking at, the DNA from a Neanderthal fossil, and the DNA is always in small bits, degraded, And so you use PCR to amplify those little bits and sequence them. So the technical challenges in both fields are very different. And in fact, it was one of the pioneers of molecular Alan Wilson, the late Alan Wilson, who was a professor at the University of California at Berkeley when we were developing PCR at CETUS. And there was a lot of interaction between our lab and Alan Wilson's lab. And it was Alan actually who realized that, hey, if you can use PCR to analyze these old crime scene specimens, you might be able to use it to analyze ancient DNA. And of course, as it turned out, that's been enormously valuable. But I would say that the first real insight into human evolution based on DNA analysis happened in Alan Wilson's lab just a couple of years before we published our first our paper. And that led to what's known as the out of Africa hypothesis. So Alan Wilson and some of his students, Rebecca Kahn and Mark Stoneking, Linda Vigilant in particular, analyzed mitochondrial DNA from living populations all around the globe. And when they did what's called a phylogenetic analysis, you make a tree of all these different sequences. And what they realized that all of the genetic diversity outside of Africa was just a subset of the genetic diversity within Africa.
And that was so interesting to me. And I had to reread some of that because I was like, how is that possible? There is a great line you have. Virtually all genes analyzed in different human populations show that there is more sequence variation or genetic diversity within Africa than outside of Africa, which is just very surprising when you first read that. And then as you think more about it and you continue reading the book, it becomes more clear that because humans started in Africa and then migrated from there, that's so important to be also studying people that have more recent African ancestry, because there's so much more rich genetic data that compared to studying people with more recent European ancestry. And that's something that I don't hear talked about a lot.
Yeah.
I mean, we've talked about that in a few episodes that I'll link in the show notes, but I thought that was just really interesting.
Well, I mean, one of the, for me, profound observations and insights into the way science works is that you can make such deep and profound inferences about human history just looking at some DNA sequence data. So basically, the idea was that there's more genetic diversity in Africa. That was the observation from the mitochondrial DNA. And by the way, this has been confirmed and supported by looking at other genes. Mitochondrial DNA actually is important both in forensics and in human evolution, because each cell has thousands and thousands of copies of the mitochondrial genome, but of course only 2 copies of the nuclear genome, one from mom and one from dad. So in forensics, there are lots of samples where the DNA is so degraded or there's so little DNA recovered from the specimen that the only thing you can analyze is mitochondrial DNA. And in evolutionary studies, it's the same thing. You have some piece of ancient DNA, and the first thing, or the most easily accessible thing you can study is the mitochondrial DNA. So before we had the whole genome of the Neanderthal, we had mitochondrial analysis. But anyway, the out of Africa hypothesis arose from observing that all of the genetic diversity outside Africa was just a subset of the genetic diversity within Africa. So the inference was that a small group of people emerged, migrated out of Africa, and eventually populated the rest of the world. This was actually very contentious. It was known as the out of Africa hypothesis, which is a phrase that Alan Wilson coined. But it was also sometimes known as the replacement theory. That is, modern humans emerged from Africa about 75,000 years ago and replaced all of the archaic human species that were present in other parts of the world. like Neanderthals or Denisovans and so forth. So, as I said, that was very contentious because there was another model of human evolution called the multi-regional idea, and that modern humans arose in all these different parts of the world independently. But the out of Africa or replacement theory was that all modern humans in the current globe emerged from a group of Africans that came out of Africa about 75,000 years ago. And when I say about 75,000 years ago, that was the other insight that Alan Wilson and other colleagues had, that you could use DNA sequence differences to estimate time differences. So that was known as the molecular clock. And so if you had the same gene, for instance, from two different species, you could measure the differences in the base sequence and using the molecular clock infer a time when those two species diverged. And the molecular clock makes certain assumptions, and I discussed this in the book because there was one issue where our lab had a difference with another lab, and I think they made this fundamental mistake. The molecular clock assumes that the DNA sequence differences that you're measuring and quantifying are, quote, neutral. That is, they're not subject to natural selection, so that the rate at which they change simply reflects the mutation rate and the population size. Because you can imagine if you have what's called purifying selection, where new variants are weeded out, that the sequence differences will be less. Or if you have what's called diversifying selection, where there's natural selection for increasing the variation in a particular gene, then the sequence differences will be greater than if it were neutral. So that's one of the assumptions embedded in the concept of a molecular clock. And as I write in the book, one of the things I was hoping to do is make clear what assumptions scientists do when they use DNA analysis. So in the forensics part, I explain what assumptions we make when we calculate a random match probability. And you may recall from the last discussion, random match probability is a metric that estimates what is the probability that, if first of all, if you have a match, you have a specific genetic profile, the random match probability is the metric that estimates what is the probability if you just pick someone at random in a given population, they would also match. And that makes certain assumptions about what we call statistical independence. And I try to make that very clear. And in the discussion of the molecular clock, and I put that in the context of a argument that our lab had with another lab, that you can only apply the molecular clock to estimate time and population size if the sequence differences you're studying are neutral, that is, are not subject either to purifying selection, which will minimize the differences, or diversifying selection, which will maximize the differences. But anyway, so Alan and his colleagues developed this concept of the molecular clock, and that's why they were able to say not only that the rest of the world was populated by modern humans who emerged from Africa, they could also say it was about 75,000 years ago.
And that it wasn't from one person. So there's this concept that you explored that, oh, we all came from, like, us as a human race came from like one couple, right? Like one woman.
Yeah.
But that is a myth that has been busted by science. Can you explain what the mitochondrial Eve is and how it got confused in the media at the time?
Okay, now that's a very good question, Kira. And I should say that it was confused in the media, but also by some scientists.
Confusing a lot of people, that even
Yes,
people that are
yeah.
working in the field. Wow. All right. So tell me about this, Dr. Erlich.
So if you make a phylogenetic tree of mitochondrial sequences, they will all coalesce to a root. And Alan Wilson referred to that as African Eve or mitochondrial Eve. And so then there was this mistaken concept that because this phylogenetic tree converged to a single individual, that all modern humans came from a single individual. But it's the nature of mitochondrial trees that they will all coalesce to a single individual because, you know, a mitochondrial lineage dies out if someone has no daughters. So if a woman has no daughters, you know, her mitochondrial lineage has gone extinct, as it were. And so when you trace a mitochondrial tree back, it has to, it's in the nature of a tree of mitochondrial sequences that would coalesce to one person. But that doesn't say anything about the nuclear genes. And so, you know, in a way, I think maybe it was a mistake to refer to this coalescent sequence, the root of the tree, as Eve, because, you know, of everything else we know. But there was another study that my lab did where we could actually estimate the size of the human population at the founding, and it was about 5 or 10,000 people. You know, it wasn't just one couple.
Yeah, because that would just change so much. And obviously there's so much in terms of what we've been able to figure out over time. But then we see in the media so many things in science that there's this concept and then the media takes it in a different direction. Or so I think this is an example of that. And you mentioned earlier Neanderthals. So is there any evidence of inbreeding between humans and Neanderthals? Because this is a concept that also gets brought up in media and different places of how closely related are we to Neanderthals? And was there a time where humans and Neanderthals could have offspring? Were we genetically close enough related where offspring could happen?
Well, I mean, it's a very interesting question, and I think the result was maybe a little bit surprising. First, when Svante Pääbo, who was the director of the Max Planck Institute of Evolutionary Anthropology at Leipzig in Germany,
And
and
won a Nobel Prize for his work.
yeah, no, you're right. Svante got the Nobel Prize for sequencing the Neanderthal genome, and I knew him when he was a postdoc in the Alan Wilson lab. And he and my friend Russ Higuchi and Alan Wilson wrote a very nice review about PCR and ancient DNA. And he used the phrase that using PCR, you could catch evolution red-handed.
I like that.
So that was a very, very nice phrase. And he also wrote a nice blurb for the book. But he was the first person even before the whole Neanderthal genome, to sequence the mitochondrial DNA from a Neanderthal fossil. And that was a very important observation because the sequence was clearly different from modern humans. So it was another archaic subspecies of humans. And so it defined another species. And then when they were able to sequence the Neanderthal genome, they found again that it was distinct from modern humans. But then when you'd sequence enough mitochondrial genomes to recognize that there were some there were sequences unique to Neanderthals, they then started sequencing or analyzing the sequence of contemporary modern humans, Homo sapiens, and they found that outside of Africa, 2%, 2 to 3% of modern human genomes had Neanderthal sequences. So that they could then infer from that after modern humans left Africa and populated Europe and Asia and the New World, that there was some interbreeding between Neanderthals and modern humans. And so, as I said, about 2 to 3% of the genomes from people outside Africa have Neanderthal sequences.
So even today, like if I looked at my genetics, it's possible that I could find some sequences in my DNA that are from Neanderthals.
Yeah, that would be the expectation. It'd be about 2, maybe 3%.
So I could be one of the lucky few there.
Well, I think it's I think most modern humans outside Africa, have some Neanderthal sequence.
That makes sense, because if it's coming from Europe and areas of Asia and different places, and then migration happened further from that it makes sense that most people that are descendants from those groups of people would then have it. Something else that I found that was really interesting, coming from a genetic counseling background, is that there were bones from 12th century that were analyzed, I'm assuming using PCR, as we've been talking about, that identified common Jewish conditions like Tay-Sachs, which totally blew my mind, because I was thinking that the bottleneck occurred way more recently that led to some of these conditions coming up, especially in the Jewish populations. But this suggests that it was way earlier than I think me and a lot of other people were thinking. Did this surprise you when you discovered this too?
Yeah, no, I discovered this really by reading. I wasn't involved in that research myself, but the fact that the incidence of the Tay-Sachs mutations was reasonably frequent meant that even back in the 12th century, when these bones came from, that Jewish Ashkenazi population had already gone through a population bottleneck. And just to be clear about what a bottleneck is, it's when a population undergoes a reduction in size, and therefore, just by random chance, some genetic variants are increased in frequency. And so that was an interesting observation, and it sort of helped time when the population bottleneck might have occurred. And as you point out, Kira, it was before the 12th century.
Yeah, way earlier
Very
than I
early.
thought.
Yeah.
Yeah, so surprising to hear that.
The other thing that's kind of relevant to the Neanderthal story is in a cave in Denisova, Siberia, there were Neanderthal fossils that were analyzed, but there was also a tiny finger bone that was PCR amplified and sequenced. And the genomic sequence that was derived was distinct from Neanderthal and distinct from modern humans. So this tiny little finger bone helped define an entirely new archaic human subspecies named after the location of the cave as Denisovans. So our archaic ancestors are both Neanderthal and Denisovan, and in parts of Asia, the modern humans, the genome sequence contains about 2 or 3 or 4% of Denisovan.
At what point in time did we make this discovery? Because I've always heard of Neanderthals and okay, aware of that. So when did we figure out this other subspecies? Do you remember kind of like what year-ish it was?
I'm guessing about 10 years ago.
Wow, so that's really recent.
And as you know, Neanderthals were defined long before DNA analysis was available by fossil remains. And there would be reconstructions. So there were a pretty abundant source of Neanderthal skulls and fossils and so forth. But there are no Neanderthal fossils, they're just little bits of bone. And it's entirely due to DNA that we have defined this newly discovered, because it's pretty ancient, this newly discovered archaic human subspecies. And not only did the DNA sequence define a new species, having sequenced his genome, we could look at modern human genomes and see that modern humans also interbred with Denisovans. And we now, especially in Asia, modern humans retain about 2 or 3 4% of the genome as derived from Denisovans.
So what is the relationship then between these three species? At what point did the common ancestor of, I guess, all three then diverge? Was it like Homo sapiens separate to Neanderthals and separate to the Denisovans? Or was it that there were maybe two branches and then one of those branches split into two? Like, do we even understand that at this point?
To be honest, I don't remember the details, but I think modern humans may have split from Neanderthals about 600,000 years ago. And I don't remember. And these time estimates would be based on DNA sequence analyses and using the molecular clock. So I'm not sure what it might have been, something similar for Denisovans, but one of the really fascinating things was not only did Neanderthals interbreed with humans and Denisovans interbreed with humans, one of the fossil remains in this cave that was analyzed by the group at the Max Planck Institute for Evolutionary Anthropology in Leipzig, they found one fossil remains where the mitochondrial DNA was from Neanderthal, and the nuclear DNA was 50% from Denisovan and 50% from Neanderthal. So this was a woman, and based on the bones, a young woman whose mother was Neanderthal and whose father was Denisovan. So that was really a fascinating observation, that was the first generation of an interbreeding event between these two archaic human subspecies.
Yeah, that fossil is like so important to find because you're seeing, as you said, the first generation, between those two different subspecies, I suppose. And as we look forward it's like we look forward but then look back. So it's kind of confusing when we talk about this, but there is this really interesting project that I've talked about on the show with Dr. Eric Green, the Pangenome Project, and how important that is of learning a lot more about the human genome and how it's different with different ancestries and different places that we are. What are your thoughts on the project? What do you hope that we glean from this project over the years?
Well, I think it's, as I'm sure Eric pointed out, a very important project, and it aspires to analyzing, in a very complete way, the genetic diversity in different human populations. And now with new technology of next generation sequencing and so forth, it is possible to generate complete genome sequences from hundreds and hundreds of individuals around the world. And I think we'll get a much clearer idea, first of all, of the extent of genetic variation, but also the pattern. And the pattern of genetic variation can tell you which population came from which ancestral group. So I'm sure you know, that most of the genetic data indicates that Native Americans, the indigenous populations of North, Central, and South America, originally came from Siberia, crossing the Beringian land bridge. That's just based on comparing DNA sequences and population genetics data from different groups.
Yeah, it's so interesting, I think, just how much we'll learn from it, and that in our genetic data, so much of it, and especially with the original Human Genome Project that was wrapped in 2003, a lot of that was based on recent European people, so having that ancestry, and so not a ton of diversity. So I think this is such a natural next step in our research to be able to capture so much more of just the diversity in the human genomes, but also we probably, as you're saying, will learn a lot more about human history too with this and migration patterns and so much within that. So I'm really excited just to see, like, as we continue to learn from it, and I'm sure continue covering it on the show. But Dr. Erlich, this has just been so fun. I always learn so much chatting with you, and I hope you're working on another book, because it's like, this was such a great read, and I always need more genetic books on my bookshelf. So that is my hope for me selfishly. But thank you so much for coming on and talking about the book, and we'll certainly link to the other episode where we talked about the first part of the book in more of the criminal forensics aspect. So thank you so much.
Well, it was a great pleasure, Kara.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.